So thanks a lot to BRCCH for the kind invitation. I'm very excited to talk about our project here. So my lab is specialized in multimodal single cell analysis method development, and we are using these methods here to actually identify B cells that produce high affinity antibodies that might be potentially neutralizing against SARS-CoV-2 infections. So first I will talk briefly about the pathophysiology of COVID-19, the different phases of disease progression. We heard about it in the previous talk as well. So what I think is really crucial is the interaction between the viral spike protein that you see here that binds to the ACE2 receptor, which is expressed on our respiratory epithelial cells. And this happens at the beginning in the upper respiratory tract. So through this interaction, the virus can enter the cell, can replicate, and can then slowly move downwards into the lower respiratory tract, into the lung, and there it actually becomes symptomatic. The inflammation leads to immune cell infiltration. We have edema within the alveoli, and this causes coughing. And luckily, this is where about 80% of the patients clear their infection. Unfortunately, for about 20%, there is this severe disease phase, which is systemic, which is mostly characterized by hyperinflammation. So in this phase, the replicating virus is actually causing the tissue damage, but in the severe systemic disease, the virus is mostly cleared, and we're just seeing the response of this over-exuberant immune response that leads to tissue damage. And here, as you know, there's the cytokine release that can cause very heavy inflammatory symptoms. There's capillary leakages. There's clotting happening within the vessels and so on. And this can unfortunately lead to a fatal outcome of this disease. So as you see already here, the immune system is crucial in actually enabling the patient to overcome the disease, or it can also cause further damage by overreacting. So what is happening on the immune level? So initially we have the virus that is recognized by the innate immune system. So for example, dendritic cells that recognize it, get activated, and then recruit the different branches of the adaptive immune response. That might be T helper cells that get activated that produce cytokines, might be cytotoxic T cells that actually kill infected cells, and what is relevant mostly for my talk here, the activation of B cells that then produce antibodies. In the long term, this might lead to memory. We don't really know yet, it's too early to tell, but hopefully there's going to be memory T cells and memory B cells that are prepared to quickly react if they're exposed to the same antigens again. So we aim to identify high affinity antibodies against SARS-CoV-2 by looking at the blood from convalescent patients as a first aim. And as a second aim, we want to study the B cell receptor repertoire across a large cohort of patients. And we're especially interested in antibody convergence. So do different B cells in different patients find similar solutions to combat this virus? And we're interested in super responders and studying their specific immunological phenotypes to learn from them. Our patient cohort comes from Basel-Land. The patient characteristics you see in the small table here on the left side. So we have a cohort of about 350 patients that have experienced COVID-19 and are now healthy again. So they had a positive PCR test, and we have 111 patient controls that didn't have SARS-CoV-2 infections, and the median age is about 50 years old. And you see here about 10% were bedridden, about 70% of the patients didn't have any restriction. And thanks to the lab of SciReady, we actually have access to a living biobank. So we have living PBMCs, or peripheral blood mononuclear cells, that were isolated for all of these 350 patients, that include, of course, the circulating B cells, circulating T cells, and that's exactly this resource that we will use as input for our screening pipeline. We also know from very sensitive serology measurements by ELISA that looked at antigens against SARS-CoV-2 that most of these patients actually show an immune response by having very well detectable IgG levels. This is from these two mid-archive preprints. Here you see on the x-axis the day or the week after symptom onset, and in the y-axis you see the level of these IgG antibodies. And you see most clearly within the bedridden patients, after about a week or two after symptoms, almost all of these patients experience a solid expression of these IgG antibodies against SARS-CoV-2, which is good for us because that means there should be B cells that produce these antibodies that we can then characterize. So let me briefly go back to the basics and talk a little bit about B cell differentiation and how the vast antibody repertoire is actually created. This might be boring to some specialists, but I think it'll be interesting for the general audience. So in the bone marrow, we have a hematopoietic stem cell that differentiates into an immature B cell. And in that process, I'll talk about it later in more detail, there's a so-called recombination of different genes that can be used to make the components of the antibody. And in the end, each of those mature B cells has exactly one antibody B cell receptor. And in the periphery, if this antibody is then seeing the antigen that it is specific for, it becomes activated. It can then become either a plasma cell that produces a lot of antibody that is secreted into the circulation, or it can also become a memory cell that can be reactivated upon future exposure to the antigen. Here, another little cartoon. So you see we have different resting B cells. They all have a different antibody. If they see an antigen that they are reacting to, this B cell becomes stimulated and it then proliferates. We have many clones. We have a clonal expansion, and all of these produce then a lot of this antibody. So what is the antibody? What is the B cell receptor? This consists of two different chains. We have a heavy chain that is a constant region, and we have a variable region, and we have a light chain that also has a constant region and a variable region. And both the variable region of the heavy chain and the variable region of the light chain together create the specificity for a specific antigen. And how does that work? Well, during this early differentiation process, we have a genomic array of heavy chain genes and of light chain genes. And you see here that's these repetitive elements. So we have this V segment that is 40 times repeated, D segment 20 times repeated, and the J segment 6 times in the heavy chain, different numbers for the light chain. And now during this early differentiation, there's this rearrangement process where one of these randomly picked segments of the V class becomes adjacent to one of the randomly picked J segments. And this is now shown here for the light chain. So this gives us about 300 different combinations of light chains. And then on the heavy chain, we also have this D segment. So here there's about 5,000 combinations that can randomly occur. So by combining any random heavy chain with any random light chain that results in the end, we get about 1.5 million options. And this is not perfect. So when this genomic recombination occurs, there is another variability at the junctions because specific nucleotides might be added or deleted, which vastly increases the space of potential antibodies, which is to about 10 to the 8th power. And then when the B cell actually is exposed to an antigen and binds to it, there's a whole machinery that specifically induces point mutations within these variable segments. So each division, this mutational machinery adds about one mutation into the variable segment. And this is about 1 million times higher than the normal mutation rate in our genome. So that's why we call this process hypermutation, somatic hypermutation. So all in all, this leads to a huge variability, a huge amount of possible B-cell receptor sequences and antibodies, about 10 to the power of 12. Now, we are very interested in these sequences because if we clone these sequences, we can produce the antibody. Now, to analyze these sequences, you need to have both the transcript that forms the light chain, as seen here, and the transcript that forms the heavy chain. Now, if you do that in a bulk sample, meaning you take a big sample and you just homogenize it, as symbolized by this fruit shake here, where all the fruits are just homogenized, you will get millions of light chain sequences and millions of heavy chain sequences, but you don't know how they're actually paired within each single B cell. That's why we need methods that preserve this pairing, because the pairing, as I showed you here 2 slides earlier, is essential for the antigen specificity. So we use a method that preserves single B cells, so we don't have to homogenize a whole bulk sample, by droplet encapsulation. So you see the methodology here. In this methodology, we use these barcoded gel beads. So we have about 750,000 gel beads, and each gel bead is full of DNA barcodes. So there's this so-called cell barcode here in green, which is unique only on this green bead. And on each of these millions of capture sites, we have this cell barcode. And it's distinct from the cell barcode on another bead. So each bead has a different cell barcode. We can then take these beads and put them into a microfluidic device, as symbolized here. And then we take the B cells that we isolate from the patient, and we feed them into the microfluidic device. And then the flow results into water droplets that are being formed within an oil carrier. And ideally, each of these water droplets has a DNA barcoded bead and a single B cell. And this bead, when we now lyse the cell, will capture its RNA and will barcode the RNA of the heavy chain and the RNA of the light chain specifically, so that we can then sequence the whole resulting cDNA and know for each of these sequences which cell it came from, so that we preserve the pairing of the VDJ sequences. Here, a little bit about the library construction. So this, I'm not going to go into it in detail, but here you see again, the bead has this barcode, which is crucial because it's incorporated into the RNA. So you see the RNA here. This is special because we actually look at the 5' side of the RNA. So we capture the RNA with a poly-T soluble primer, and then we do a reverse transcription, and the reverse transcription adds at the very end these non-templated Cs. These are captured by the primer that sits on the gel bead. And then we have a double-stranded nucleotide, which has in the cDNA part here incorporated the mRNA transcript, and here the cell barcode. So we know from which specific cell that comes from. So this method enables us to use droplets as mini reaction vessels, so that we can profile about 10,000 B cells in one small reaction vessel, and then know for each one of them which cell they came from, and we can pair the heavy chain and the light chain information, and of course get many other modalities that we want to measure. So here we will then use a nested primer system, a nested primer that binds in the constant region of the antibody encoding transcript, and amplify the whole VDJ full length, both for the heavy chain and the VJ sequence for the light chain of the antibody. That's great, but this technique is very expensive, and to get to 1,000 cells can cost us thousands of dollars. So this is not applicable to this large cohort of patients that we have. So we need to add another equipment or another piece of technology to make it feasible to go for a lot of patients. And this is a system that is called cell hashing. So cell hashing enables us to label cells from different patients and then to pool them and still do single cell analyses, and at the end to know from which patient they came from. So you see here 3 different cell populations that could be from a patient A, B, and C. These get labeled with a DNA barcode that is here bound to an antibody that looks like a RNA, has a poly A tail, which means if we put it into this microfluidic workflow, we will then also transcribe that into cDNA, and we can actually read it out when we're sequencing. So there we can now pool all these samples and just treat them as one single sample. So we can take cells from 10 patients, put them together, run them into the microfluidic pipeline, and then at the end, sequence the library from the gene expression or from the VDJ light and heavy chains. And because they have different PCR primers, we can also sequence this cell hashing library and get for each cell information if it came from patient A, B, or C. We actually don't use an antibody-based system. We use a lipid-based system. It's called multi-seq, that is based on a lipid anchor here that integrates into the cell membrane, and that is bound to a DNA barcode, which can then be captured and used for single cell genomics with exactly the same outcome, that we can pool many different samples. This is a bit cheaper, and here we can actually pool and we can quench the reaction, so we don't have to wash and lose valuable B cells. Great. So now we can pool many different samples. Now in the next step, we also want to know if these B cells are actually reacting to antigens of interest. So we heard a lot about neutralizing antibodies. Ideally, we want to get antibodies that can block the entry of the virus into the cell. So for example, antibodies that would block the receptor binding domain of the spike protein and then cause them to not being able to bind to the ACE2 receptor on our respiratory epithelium. So for that, we have been developing a method that was recently also published by another lab as under the name of BRCCH. And in this method, we use a bait. So you see here the bait. The bait is consisting of an antigen, for example, the spike protein or the receptor binding domain of the spike protein, any domain that is very likely to generate neutralizing antibodies. And this is then fused to a DNA barcode and a fluorophore or a tag, which enables us to rapidly pull out these cells. We can then incubate this antigen with the DNA barcode with purified B cells. Now here, this is mostly relevant for memory cells because they can still have B cell receptors which are bound to their cell membrane. For most of the plasma cells, this is not relevant because they don't have any cell-bound antibody anymore. This is just secreted. But memory cells that have an antibody or B cell receptor that can bind this antigen would then be bound by our bait. And we can then use enrichment methods, for example, flow cytometry or magnetic sorting, to specifically isolate cells that have a lot of the baits bound. And then we can take this whole construct and put it also into the microfluidic pipeline, because the barcode again has a DNA barcode part and a poly A tail so that it looks like an RNA, which means we can then actually create a third library where we see for each cell how many of these baits that it bind to. And the beauty of this system with the DNA barcode is as well that we don't have to focus only on one antigen. So as you see here in the SARS-CoV-2, we have 4 different proteins. We have the membrane protein, nucleocapsid protein, spike protein, and envelope protein. And mostly the spike and a little bit the nucleocapsid protein are thought to give rise to neutralizing antibodies. So we can then take different antigens and create a library of antigens. And we can identify which antigen we see because we can attach different DNA barcodes to each antigen. So we can actually incubate our B cells with a whole library of different antigens, and then by sequencing measure actually how many of which exact kind were bound to this B cell, and then of course look at the resulting VDJ B cell receptor in this cell. So our pipeline. We have our cohort of the living biobank of these PBMC samples. We first use sample hashing so that we can pool 10, 20 patients together. And then we enrich plasma cells or memory cells. So that's, again, cells that produce a lot of antibody, and that's the cells that have these surface-bound antigen B cell receptor sequences that could be reactivated when we are seeing the virus again in the future. And then we bait antigens by using a library of epitopes that might lead to potentially neutralizing responses. And then we load the whole thing onto this 10x droplet encapsulator. And this gives us the multimodal characterization of about 10,000 single B cells. So for each cell, we can learn the paired VDJ sequence of the heavy and the light chain to then also clone the antibody if we want to. We can get the transcriptome and learn about the identity of the B cell, about its state of the transcriptome. We learn about the sample origin, which patient is all of this coming from. And then, of course, we can look, was that a patient with severe symptoms, not so severe symptoms? And we can also look into the serology and see what kind of a response did we measure when we looked in the serum in terms of IgG and in terms of neutralizing titer of that specific patient. And crucially, we can now, as I explained here, determine the identity of the bound SARS-CoV-2 antigen and its quantity. And of course, then we can express promising candidates, test neutralizing antibodies in vitro, and also look at their neutralizing potential, maybe with collaborators in animal models downstream. So what's the therapeutic potential of a monoclonal antibody? Here you see a scheme from this review paper. If you inject a patient or a healthy person, the hope is that you can block with a neutralizing antibody this interaction between the spike protein and the host cell, the ACE2 receptor. This might be helpful in healthy patients that have a high risk because they just had a recent exposure to an infectious patient, or because they're working on the front line in the clinics, you name it. It could also be helpful in patients that are in the acute phase of the disease to help their immune system. So it has maybe an application within prevention and also within treatment. Of course, there's big companies doing this already. So there is 2 ongoing phase 3 trials, randomized controlled trials, one from Regeneron that is using a mix of antibodies. You see the clinical trial registration, and one from Lilly that uses another mix of monoclonal antibodies against SARS-CoV-2. What do I think are the big challenges? I talked a bit about it in the beginning. So here you see again these important clinical phases of SARS-CoV-2 infections of COVID-19. So initially we have this viral phase where the damage that is caused comes from viral replication. But shortly thereafter, the viral load actually decreases. And now the damage is caused by the cytokine storm. And further on, we have this hypercoagulation and unfortunately these long hauler symptoms in some patients. So here, in terms of drugs, there is now clear evidence that steroids are helping. Here, there's not so much trial data, unfortunately, yet, but still there's a lot of, it seems that the blood thinners are doing well in this coagulation phase. And I think it's very important to determine the therapeutic window for antiviral therapies, because likely they will only work at the beginning in the viral phase when the virus is actually present and causing damage. And I think that's very challenging also for example, remdesivir. How do you check this appropriate therapeutic window, and how can you get this drug to the patient? Because monoclonal antibodies usually need to be injected intravenously. And the same is true for remdesivir. But as you have seen in the beginning, in this first phase, in this first week, the patients are usually not hospitalized. So that's of course very challenging, and I think future research is needed in formulation. So in oncology, there is some monoclonal antibodies that can be injected subcutaneously. That might be more feasible in an out of hospital setting within the viral phase. So I think therapeutic window for antiviral therapies, that's going to be a very important factor to determine for successful therapies. With that, I'd like to thank my lab members, the collaborators, Iredi, Fabian Rudolph, and of course, Botnar Research Center for Child Health for enabling us to do this work.